Welcome to today's webinar, Immunophenotyping in FCS Express 5. Thanks a lot for joining us. My name's Kaya Ghosh. I'm an application specialist with De Novo Software. On the line, we also have our Director of Sales, Tanya Tolmachov, and she will be answering any questions that you type in here where it says enter a question for staff. So any questions that come up, just feel free to enter them in, and we'll either answer it right away or we will follow up with you later offline. So feel free to enter anything in there. That's what makes this webinar interactive and interesting. So what we're going to talk about today is a basic immunophenotyping workflow. And the purpose of this webinar is not really to go into a great deal of detail on any particular one function, but rather to give you sort of an overview of the general types of functions that you'll need to use in order to create a basic layout, which we call an immunophenotyping layout. So basically, in a nutshell, what we're going to cover is loading a layout that's pre-existing with data files. It's a partially built layout, and then inserting new plots and gates, creating default and custom defined statistics, batch processing to PDF, PowerPoint, and Excel, and reusing a layout as a template. Okay, so some of you are probably already familiar with some of these functions because you may use pre-existing layouts in your clinical or your research labs. So you probably have an idea how to do some of these things, but hopefully we'll cover the basics as well as showing you a few interesting tidbits throughout the course of the webinar. So there'll be something for everybody to learn, regardless of your skill level. So I'm going to get started here, like I said, with this partially built layout. And this layout actually does exist in your tutorial sample data folder, which I'll show you how to download later, so you can play around with it and kind of replicate everything that we're going to do today. So far, we've got some empty plots because this layout was saved unlinked. So to get the data to show up, of course, we're going to load up the files by coming to the Data tab. And there's a couple different ways to do things. I'm just going to show you one way. Don't worry if you're doing it a different way in your lab. That's totally fine. But I'm going to use the Data tab and the Select button to load up the files that we want to put in. So we're going to put in this sample1 file. Okay, and you can see that if I open up the data list from the Data tab, we've already got the other FCS files that were in that Windows folder loaded into the data list. So there were 4 files, and as soon as we loaded up one of these plots with the first file, the other 3 files came along. And so those are ready for us to analyze as we go forward. Okay. So what we're going to do now is insert a new plot. Some of you might be aware there's different ways to insert plots. You can come to the Insert tab, for instance, and put in a density plot or any other kind of plot. Let me undo that. Another way to do it is to drag from the data list onto an empty spot on the layout and select the plot type you want, and there we have a nice contour plot. And yet a third way, which is what we're going to work with today, is inserting a plot from a gate. So this scatter gate is what we want to drill down on. Typically you have several layers of gates in your hierarchy, and so this is the highest level gate that we want to focus our analysis on. So to put in a scatter gate, I'm just going to drag that out to an empty spot on my layout, and now I've got a new plot gated on scatter. Okay, so let's change these parameters to something a little more interesting. Okay, so now we're looking at CD19 on the y-axis, CD45 on the x-axis. So let's take a closer look at our CD19 positive cells. So I'm going to go up to my gating tab and draw a gate around my cells of interest. Okay, and as soon as you close that gate, you're faced with this create new gate dialog. So we can give our cells a name here, maybe change the color. Okay, so, and you can see that these are, since these are color dot plots, we have some nice backgating information already displayed. And what I mean by backgating is when you draw a gate on one plot, you can see where those events fall on a different plot in a completely different parameter space. So this B cells gate was drawn here on this plot on the right-hand side. But if I take a look at the left-hand side plot, you can see that we have the green dots appearing here in this scatter plot here. Now, if we take a look at our gate view, you can see that we have two different levels of gates. And you can play with your hierarchy here. So right now, B cells is dependent on scatter. If I wanted to drag the B cells out to beyond the first node, you see now B cells is independent of scatter. And it might be hard for you to see. Let me highlight these cells. But some of these green cells now, they're not restricted to that initial scatter, that initial scatter gate. So here's one green B cell. Here's a couple up here. Okay. Okay, so let's put that back. But that just shows you some of the flexibility in creating your gating hierarchy. Okay, so let's take a look at this other plot that we have down here. So on this plot, we are looking at a couple of different other parameters. We're looking at IgM and IgD, and we can see the backgating information here, the green and the red, but it's kind of hard for me to figure out where to put my quadrants, especially if I kind of go back and forth through the files. You can see that that boundary is harder to see on some of the other files. So in this case, it might be useful to look at a different plot type rather than color dot plot. So I am going to reformat this plot into, let's say, a contour plot. Okay, and maybe I'll even put in some of the outlier dots, and this is an important section right here. If you right-click on any plot and go to Format, you get this handy formatting window where you can change any kind of formatting aspect of the plot. So let's put in those outlier dots, and I'll do a preview. Okay, so you can now see that's going to be a little bit easier for us to judge where to put the quadrants. And any kind of formatting you'd want to do is available here. So right now in our title, we just have the FCS file name, but I can put in, for instance, the current gate of the plot. Okay, now you see that it's not gated. Okay, quads, if I wanted to not show them, I could uncheck it. Okay, or I can recheck it and say, you know, standard if I want to do the standard quads. So basically anything that you want to do is there. Okay, so let's put that back on floating so I can adjust these vertices how I want them to look. Does anybody have any questions about what I have done so far? Okay, don't forget, if you do have any questions, for those of you who have logged onto the webinar a bit late, the questions and answer box in your GoToWebinar control panel here, where it says enter a question for staff, feel free to type anything in there. All right, so right now we are looking at an ungated plot here in the lower left, but we really want to focus on our scatter cells and our B cells. So let's apply a gate to this plot. And that's very easy to do. There's lots of drag and drop. So the easiest way I'd say is just grab this B cells gate from the plot that it was created on and drop it onto that plot. So now you see that this plot has been gated on B cells. So you can see that there in the plot title. Okay, you can also gate plots from the gate view here, which is the interactive legend of your gates. So if I wanted to drag the scatter gate onto here and gate it on scatter, that will work. And lastly, this gating tab is very useful because it has this current gate drop down here, and you can use that to simultaneously gate different plots. So I can do a control click and highlight both of these and say that I want to gate both of these on no gate, for instance, or B cells. Okay, but let's keep this one gated on scatter. Okay, so those are the ways that you can apply gates in FCS Express. It's fairly intuitive. All right, so let's put in let's put in another plot. Okay, we'll put in a histogram this time. So that's going to be here on the Insert tab. We can take this histogram and draw it to the size that we want initially. And let's change the parameter to something a little more interesting. Okay, so we're looking at CD20. And let's say we actually want to look at the CD20 expression of our double positive and our double negative cells from this plot at left. So we've got some IgD, IgM positive cells here in the upper right quadrant. And we also have some double negative, IgM negative, IgD negative cells here in the lower left quadrant. So normally quadrants in most programs, they're only applicable to 2D plots, but in FCS Express, you can actually apply these quadrants to a 1D plot indirectly by creating a gate from the quadrants. And so we call that converting and linking a quadrant to a gate. So that's going to be via this handy right-click menu, which I've shown you before. We'll come over here to convert quads to gates, and you see we have the choice of converting individual quads or the union of two quads. So what we're going to do is convert and link the upper right and we can give this gate a name. So I'm going to call it the upper right quad, or you can actually call it something a little more descriptive, like maybe double positive. It's your choice. Okay. And now you see up here in the gate view in the upper left corner, we have a double positive gate that's a child of that B cells gate. Now I'm going to drag that double positives gate onto the histogram, and so now this histogram is gated on double positive. Okay, but let's create an overlay. Like I said, we're really interested in comparing the CD20 expression between the double positive and the double negative cells for IgM and IgD. So let's go ahead and convert and link this lower left quad into a gate as well. Maybe I'll give these a lighter blue color. Okay, so now the question is, how do we create an overlay? Some of you might already be used to creating overlays in FCS Express, and so you know that it's easily done by drag and drop. There's a couple different ways we can do it. One way is to just drag one plot into another plot, and you can kind of hover over and then say, add the files to the plot as a new overlay. And then we have two overlays there. They're both gated they're both gated in the same way, and that's why you don't really see two overlays. I'm going to undo that and show you one other way to create an overlay, which might be more intuitive for you if you're used to looking at the data list, and that is to just drag the sample from the data list onto the plot. So I'm going to grab my sample 1 and drag it there onto the plot. And again, this part is identical. We're going to say add the files to the plot as new overlays. Okay, so it turned red. So we actually have one black overlay and one red overlay. But let's format these overlays so that they actually take on some different colors. We'll add some smoothing, and most importantly, we'll apply different gates to each overlay. So that's most easily done via the Format menu as well. So I'm going to come over here to my Overlays category. And the first overlay is already gated on double positive, so that looks good. Let's actually give it a color so it matches the gate. We created a royal blue double positive gate, so let's assign that overlay that nice royal blue color. And now I'm going to come over to my second overlay, and that we gave this light blue color here for the double negative gate. And I'm going to give it a light blue color as well to match. And most importantly, we're going to assign the gate to the second overlay to be double negative. And that's right up here. So everything's here in the overlays category. Now I can do a preview, and you can see clearly we have two different overlays there that are gated differently. FCS Express doesn't smooth histograms by default, and that's why the data looks a little bit, I guess, noisy or ragged, you would say. But you can certainly apply smoothing. Just highlight both of your histogram overlays here. And maybe we can apply a modest smoothing of, say, 6. Okay, so now you see that the data takes on a smoothed look. Don't be afraid to use smoothing. It doesn't affect your statistics. It's strictly a display option for convenience. And you can increase the line width here as well. So if you want to make them thicker lines. Okay, new in FCS Express 5, we have this nice transparency feature, which works for filled histograms. So if I just fill the histograms, it looks like that. But we can put in, let's say, a transparency value of 35. And now you can see that when you have overlapping populations like you do on this histogram, that you can sort of see through each layer a little bit better. So that's really actually very useful if you're going to have 5 or 6 overlays and there's not a lot of blank space on your plot and you really need to see each peak. That's where that transparency comes in handy. So we'll leave that there. Another thing you might be wondering is, why is the count on the y-axis 44? And why do we look like we have such a different number of events? Well, that is because if you take a look at the original quad that the gates are derived from, we have far fewer cells here in the lower left corner than in the upper right corner. And so that's why this light blue double negative gate shows up as a much less abundant population here on the histogram. The peak height is lower. Okay, and so that's an unnormalized histogram. So we don't normalize by default, but we can apply normalization to our histogram if you'd rather see your peaks being at about the same height. And so that's here under the histogram specific options. I'm still in the formatting menu that we had open before. And we're going to put in a normalization to, let's say, a peak value of, we can do 100. So keep your eye on the histogram when I click preview. Okay, so now you see that those have been assigned the same peak height. And again, this is just like smoothing in the sense that it doesn't affect your statistics. It's strictly for display. So feel free to use whatever kind of normalization options you need to use to see the data in the way that you need to see it. And there's other normalization options as well. So I normalized to peak value, but you could normalize to number of events. And I'll leave it on sample 1, for example. Okay, so now you see these have both been normalized to about 44 events there on the peak height. Okay. Do you all have any questions about any of these formatting options that we've applied to the histogram so far? Okay, well, if there's no questions, I'm going to click OK to accept the change. Okay, so we've actually made it quite a ways. Just to point out where we are in the workflow, we have successfully loaded a layout with data files. We've inserted plots and gates, and now we are ready to create default and custom defined statistics here. So this is very easy in FCS Express, but hopefully I'm going to show you something that you may not be aware of, and feel free to ask any questions at any time. Okay, so you're probably all aware that you can add custom statistics very easily simply by right-clicking on a plot and going here to statistics in that pop-up menu. In this case, I have a choice between gate statistics and quadrant statistics since I have both on this particular plot. I'm going to put in the quadrant statistics. And let's move this down so it's not covering our plot. And now I can format the statistics window to contain the elements that I want and to get rid of the elements I don't want. So simply by right-clicking on the statistics window, I can take away overlay number. We don't need that. Maybe I'm going to take away percent of all cells, and let's add in X median. I also wanted to point out that new in FCS Express 5, we have these BD style stats that were requested by some of our customers. So if some of you out there are used to using the BD, RSD, or RCV, or these percent of gated parent, percent of gated grandparent stats, these are here for you in FCS Express now as well. Okay? So let's move these over. I want to make this fit nice and neat on the page. Let's see if we can do that. Okay, maybe I'll take out the file name. That way everything's a little more compact. Okay, so all of these statistics are going to live update, so you can watch them updating as I'm moving around the quad. Note the backgating also updating on the two plots in the upper part of the layout. So FCS Express 5 has this live updating gates. I don't even need to let go of the mouse. I'm just continually readjusting my quads, and everything is updating in real time. All stats, all plots, all gate colors. Okay, so that should really be helpful to you in placing your gates. Okay, so that is pretty useful, adding these default gate statistics and quad statistics windows. But keep in mind that the statistics here in this window only pertain to this plot here. So this plot is what I right-clicked on to insert the statistics window. And so all of these statistics only pertain to the B cells because this plot here happens to be gated on B cells. You can see in the title of the plot. If you want to insert statistics from an ungated population, i.e. the entire FCS file, what you can do is just right-click on an ungated plot, such as this plot in the upper left, and put in your gate statistics. And there, these statistics, such as the number of events here, are going to reflect the entire FCS file. Okay? But usually when you want to summarize an experiment, you want to draw in statistics from multiple plots on your layout. And I know some of you are clinical customers or research customers who are using very complex layouts, sometimes with multiple pages, 5, 10, I've even seen 50-page layouts. And so you can imagine in a layout like that, one of these gate statistics windows, it's not really an effective way to summarize the entire experiment. So in that case, what you want to do is insert one of these nice custom text tables, and we have one already inserted here. And what we can do here is pull in statistics from multiple plots on our layout. Okay, so this is already nicely formatted. You can see the coloring and the filling of the cells, but just really quick, I'll show you how you could put one in anew. It's very similar to how you would do it in PowerPoint or other Microsoft Office programs. You just come up to the Insert tab, put in a text box anywhere on your layout, and then you can right-click within the text box and go to Insert Table there from the pop-up menu. Okay, so there we have a table. All right, so that's already been done. So we're going to use this really pretty one that's already created. Okay, so let's populate this first column of statistics, which is the mean FSC, the mean forward scatter values, and we're going to do that by dragging and dropping. Okay, so we will put in a statistic by dragging and dropping this plot down into the table. You see my cursor becomes a little plus, meaning that the cell is ready to receive a statistic. All right, and it's asking me what I want to put in, so I do want to put in the statistic. So I click OK. And what we want to do is this first row in our table pertains to the double positives, IgD positive, IgM positive. So let's select that here from the gate dropdown, double positive, and we're going to put in the X arithmetic mean. So the X arithmetic mean from this plot that I've highlighted is essentially the forward scatter mean, because that's what's on the X parameter of the plot. Okay, so you can probably see that this statistic is now highlighted in gray. And the reason for that is because it's a live updating token. So I can't edit it directly. You see, if I try to edit it, I don't really get a cursor because it's actually a statistic that is drawn from the plot. But if I click outside of the text box so that it's no longer in edit mode, you can see that it is normal text. And I'll just zoom in so it's a little bit easier for you to see on your computers at home or at work. Okay, so you can see that that's about 136,000. But if I move that quad around, you can see that it's updating, right? So that's what I mean by a token is a live updating statistic. Okay, so let's populate that other cell right below it as well. And I'm just going to do it in the exact same way. I'm going to drag this down into the cell. At this time, I'm going to select my double negative. And we're going to put in, again, the X arithmetic mean. Okay, and so those are slightly different. Those double negative cells are slightly smaller on average, as measured by forward scatter. All right, let's do the same thing now for the second column of statistics in this text table. So this second column is entitled CD20, and maybe I'll just zoom in a little bit here. So what we want to look at is the CD20 expression for our double positive and our double negative cells. Okay, and we're going to grab those statistics here from this histogram. But first, let's draw a marker so we can delineate which are the positives and which are the negatives. So you can do that here from the Gating tab. You can grab a marker, or you can just right-click and do Create Marker. So hopefully you're getting a sense there's usually multiple ways to do things in FCS Express. Okay. So now I've got a nice marker, and that's going to update, of course, as I move that around. All right. So let's drag this plot into that cell so we can put in that CD20 statistic. And this is much the same procedure as we've already seen. So we're going to select M1 from the marker because our M1 is our CD20 positive cells. And the first row of stats is going to be drawn from our double positive. So I'll click OK. Oops. Looks like I didn't quite click it. We want to do percent of gated cells. There we go. Okay. And now I'm going to show you a shortcut. Rather than just dragging the plot over and over into the cells, what you can do is use the clipboard to copy paste and then just change the gate that the token refers to. So I'm just highlighting this with my cursor, and I'm going to copy it. So you can do Control C or right click, whatever you want. And now I'm going to come down into this cell right below it and do a paste. Okay, so right now these are identical. They're both 98.87. That's because I just did a copy paste. But what I can do is format this token here in this cell here that I've highlighted to refer to the double negative gate, and that way we'll have two different statistics. Okay, so that's as easy as you could right click and go format token, and I'm going to come over here to statistic, and so you see everything is basically already defined for us. We are already getting the same stat, percent of gated cells from M1. All we need to do is change this to double negative. Okay, so that is coming up as 0 right now. I think we just need to format this marker to apply to all of the histograms. So we'll come in over here. Oh, I know what it is. Let's go back and reformat this token. There's one thing I forgot to do. So we did select the gate. What we need to do is come over here to data source and say that we want to get that statistic off of the second overlay, the light blue overlay. Okay, so that's data source, and we'll select the second overlay, and now we have a value. Okay, and that's going to update, of course. Both of them will update as I move this gate around. All right. So those are some nice default statistics that we've put into the table. You can imagine these tables can get as complicated as you need them to be. We have some customers that have tables with many different columns that kind of span the whole page. You can certainly do that. This is a rather simple illustration. But let's do one more cool thing. We're going to create a ratio of the CD20 expression between the double positives and the double negatives. So you see that we already have a cell here dedicated to that ratio. And that's a custom defined ratio. That's not existing by default in FCS Express, but we can create that ratio in FCS Express using custom tokens. Okay, so that's here on the View tab. I'm going to open up my custom tokens, and this is going to work very similar to Excel. So the syntax is going to be quite the same. So let's just create a new token. I'm going to call it ratio. And in Excel, you would create a ratio by typing equals and then dividing one cell by another, right? So we could say equals 20 divided by 5, and then I can drag this ratio out anywhere on the layout, and you see that it's been evaluated as 4. I can drag it, for instance, into the cell, and it's evaluated as 4. Okay, of course, that's not really the statistic that we want. We don't want to just divide one number by another. We want to divide the number of events in this marker for one gate divided by another gate. So let's delete out these numbers, and this is going to work really similar to what you've seen so far. So just dragging and dropping to create statistics. You can do the same thing here in the custom tokens window. So I'm going to drag over my plot and say OK. And we're going to select the double positive number of events. OK, and let's just expand that. So right now you can see that that is highlighted in blue because it's a token, and it's basically the number of events for the histogram in that first overlay. If you're curious what that value is, you can change the display text to value. Okay. All right, let's just check one thing here. Forgot to select the marker. There we go. Okay, so that should match up now. And now I can type that divided sign, and let's drag. We can drag the plot over, or we can take another shortcut. We already have that statistic defined here in the text table. So I will just copy it and place it in here. Okay. So now you can see that has been calculated as a ratio, okay? But I basically divided 2% by another. To make this really correct, we should really divide number of events each time. So I'm going to change that to number of events. It's really up to you. Generally, number of events is better to choose because that way you don't really lose any significant figures and it's just less confusing. You're just dividing one raw value by another. Okay, so there we go. Okay. And so now you can drag this around and you can see that everything is updating. All right, so we're almost ready to go here. We've created some statistics, and now we're ready to move on to the next portion of the webinar, which is batch processing to PDF, PowerPoint, and Excel. Okay, so batch processing tools are here on the Batch tab. We're going to click on Batch Actions. And these are floating windows, so I can kind of move them around and mouse over this icon in the center to sort of tuck this away on the left. And let's delete that text table that we don't need. I can even grab my data list and sort of tuck it inside, so now everything is nice and neat on my layout. Okay, so one thing we could do is add a print, and you can choose to send it to any printer that your computer is hooked up to. And that's handy if you just need a quick printout for your lab notebook or something like that, because FCS Express can communicate directly with your printer. All right, and we're going to also add a PowerPoint action. And this way we'll have an external record of our entire data set in PowerPoint. So I know this is a rather large window. There's only a couple of things you need to pay attention to, and I'm going to outline those here with my pen. One of them is pretty obvious here. It's the output file options, where you need to define a save path and a file name. And the second one here is iteration options. Okay, so iteration options refers to whether you want to have a separate PowerPoint file for every FCS file in your data list. So we have 4. So we would, in that case, have 4 different PowerPoint files. I think in most cases, if you're doing a batch process for an experiment with multiple files in it, you probably want to just combine them into one and have one big PowerPoint file. Okay, and let's define our save path down here. So we're going to put this on our desktop and we'll call it our immunophenotyping webinar. And we'll click OK. All right. We can do a PDF as well. It's quite similar. We can choose high quality, for instance. And let's put this on our desktop also. Okay, in this case, let's actually save a different PDF for every FCS file. So we're going to end up with 4 PDFs. So I'm just going to leave the iteration options on, save the output of each iteration separately. If I click OK, you see I get this error message. It says, since you have not specified a token in the output file name, all iterations will have the same name and will overwrite each other. That's because we can't really have 4 different files that are all called ipwebinar.pdf. Windows won't allow that, correct? So we're going to correct it by adding a suffix to the file name that will change for every single iteration. So you can click on T for token. The easiest thing to do is probably to come down to batch processing and click on current batch iteration. That way your PDFs will be named IP webinar 1 1.pdf or 2.pdf and so on for the number of iterations you have in your batch process. So that's definitely the easiest thing to do. If you want to get a little bit more fancy and a little bit more descriptive, you can click on keyword, and we can actually pull in a keyword from our data files. So if I come down to $file, this is now going to be called IP webinar and then the file name for the current FCS file, so sample 1. Okay, some of you are already familiar with keywords, so if you're using things like sample ID or patient ID, things like that, you can pull that into the file name as well. All right, so let's click OK. So these are going to be our visual outputs. Lastly, let's put in an Excel. So Excel is handy for if you want to collect all your statistics in a spreadsheet, obviously. We did a lot of calculations here in FCS Express together, so I'd like to encourage all of you to do as much of your work as you can in FCS Express, because then the statistics will automatically update when you move your gates. But of course, there's some cases when you might be collecting a whole lot of statistics, and maybe you need to do a downstream analysis in another program, or just need to keep an Excel spreadsheet for quality control purposes. That's what exporting to Excel is useful for. So we'll set up one of those together. So click on column mode, and we're going to put this again on our desktop. Okay. So now we just need to tell FCS Express which statistics to export. So we don't export all of them by default, because then you would have basically this looks like 15 or 20 statistics for every single gate in your layout and every single plot and every single file, and that would quickly become too large of a spreadsheet to make sense of. So the way FCS Express is set up is that once you are done building your layout, you decide which stats you want to export on the fly. Okay, and so you can do that by dragging and dropping, you probably guessed. So if I want to drag, for instance, this upper right number of events, I can drag it from the statistics window to the Excel, and there it is. You can also drag gates. So if I want to drag this B cells gate over, then we just need to tell the program what we want to look at. So let's look at the Y geometric mean, so we can get the mean of the CD19 expression. All right, this nice custom table that we built together, we can drag all of the stats over from it simultaneously by grabbing it from its border. You see my cursor becomes a little four-headed arrow. Okay, so you see all 4 of those were dragged over simultaneously. Okay, and the last thing we will do is put in a keyword. So right now, each one of these statistics is going to come through to the Excel, and that's nice, but we also need a descriptor to know which file we're looking at for every single row. And so the best way to do that is to drag over a keyword. So we're going to do that by dragging over a plot. And you've seen this dialog window appear before, right? Because we put in statistic tokens. This time we're going to put in a keyword token, and that's going to allow us to put in any kind of descriptor from our files. Okay? So these keywords are metadata that were acquired with your FCS files when you were on the machine. So you're always going to have this $sign file. That's a failsafe. You definitely have a file name. But for those of you who are familiar with keywords, you might be using things like sample ID, tube ID, specimen ID, or maybe SMNO, depending on what instrument you're using. And so these are all going to be down here. As you scroll, you'll find the keywords that you need and that you're accustomed to using. Okay? I'm going to keep it simple for today and just use $sign file, because that's what everybody has for sure. Okay, so I see keyword is now last in my list, and if I were to export this, that would be basically the last column in my Excel, but I really want that descriptor to be column A in my Excel. So I'll just drag this up here, And so now that's going to be my first column in my export. Okay. Does anybody have any questions? Okay. Before I click Run to run the batch, let's just take a preview of the samples that we see so far. So here on the Data tab, you can kind of scroll through using the Next and Previous buttons and see what all of the files in your data set look like. So I'm going to go to the next file. That's sample 2. Now we're looking at sample 3. Now we're looking at sample 4. So that all looks pretty nice. So I think we can go ahead and run the batch. And then maybe we'll come back and do a couple of adjustments, depending on how it looks. So the quickest way is just click on the lightning bolt run. So that's going to generate very quickly. And I hear my printer going off in the background because I set up that print action. All right, let's take a look at the Excel first. Okay, so you see that we have 4 rows of data, and that's because each row or each iteration is its own row in the Excel file. Keep in mind, this is a really simple example we've worked with today. I know a lot of you out there might be using panels where you have, let's say, for every patient, you have 5 different tubes. In that case, you won't really have a one-to-one correspondence with the number of FCS files and the number of iterations, and that's totally fine. That's very frequently the case, but we're just setting up a very simple example today, just for the purposes of illustration. So, and then you, of course, have one column for every single statistic that you chose to export. And you can take a look at the last column over here. Here's that ratio that we chose to export as well. Okay, so I'm going to close that. And let's take a look at some of the other exports that popped up. So here is our PowerPoint export, and we have 4 slides you can see on the left-hand preview, because we had 4 different iterations or 4 FCS files. And the nice thing is that this PowerPoint export looks exactly like FCS Express, right? You almost can't tell what program you're in because it's basically an identical export to your live layout in FCS Express. But the thing to note is these are individual objects and you can move them around and resize them if you need to, okay? There's sample 2, there's sample 3. And if you ever have multiple layouts open, and you need to cut and paste using the clipboard, you can do that as well. So I can grab this picture from another layout and put it inside of this PowerPoint. It's a little bit on the large side. Maybe I'll make that a little bit smaller. There we go. Okay. And lastly, let's take a look at our PDFs that popped up. And they are here. I'm going to drag them over to my desktop so you can see them. Okay. So here's the other one. Okay, so now you see that these were appended with that file name that we put inside of the file name using a token. So there's the IP webinar sample1.fcs.pdf, there's sample 2, sample 3, sample 4. Okay, so we can open up any one of these and just take a look. And you see that this again looks exactly like your layout. So all of the statistics, all of the objects that you had in your layout, whether they're plots, stat windows, pictures, they're all going to come through looking exactly as you intended them to look. Okay, so let's close that. I'm going to show you one more thing. We do have a few more minutes. Sometimes when you're running a batch process, you need to adjust the gates between samples. So in this particular layout, for instance, I might want to adjust my quadrant boundaries. So you see this quadrant boundary for my lower left plot looks good. When I go to the second iteration, maybe that's not so good. I need to adjust that, right? Maybe I would want to adjust the marker, maybe adjust the B cells gate. So the best way to do that is in a batch process by adding a pause. Okay, so we're going to come over here to our batch options, and we'll put in this checkbox, unconditionally pause between iterations. All right, and as soon as I run this, I think you'll see exactly what I mean. It should be pretty self-explanatory. So I click run, and it's making those 3 exports, so 3 of 3 actions executed, and we've only done 1 of 4 batch iterations, or one of 4 FCS files, and it's basically giving me the chance to adjust the gates. So this sample 2, I think this B cell gate, we can make it a little bit smaller. Okay, maybe we'll adjust the quad a little bit. It's kind of hard to judge where that one should go. All right, I can make the marker a little bit more restrictive too. All right, and now we have the choice if we want to keep these changes or restore the state of the layout before the next iteration. So let's restore to the baseline, which is the second radio button here. And I'm going to click continue. Okay, and now you see the B cells gate went back to its original position, and so did the quad, and so did the marker. Okay, so let's click continue again, and maybe just for illustration, I'll make this scatter gate really large, just so the change is very obvious on the PowerPoints. Okay, and I'm going to click run to end, because if you have a lot of samples and you're pretty sure that the rest of them are fine and you don't want to go through them one by one, you can just click run to end to finish off the batch. Okay, so there's that Excel that popped up again. You'll just have to take my word for it that the statistics are reflected in there according to the gate changes we made. But that should be pretty obvious when we take a look at the PowerPoint here. So this is the first iteration with the original gate positions. Now I'm going to go to the second slide, and you see that that B cell gate was made smaller. We moved the quad over a little bit. We made that marker a little bit smaller. Go to the third iteration, everything went back to baseline. And then the number 4, I made that scatter gate very large. Okay, so that should be pretty useful for you if you need to adjust gates between samples. It allows you to do it very quickly, make the decisions as you go. And I just want to point out that in FCS Express 5, if you were to save, close, and reopen this layout, you could click rerun here rather than the run lightning bolt, and then these batch processes would be re-exported with all those gate changes that you made in FCS Express, okay? And that's a feature called Snapshots, which we do have a webinar all about. And if you have any questions about that, feel free to type that in. We can definitely point you to the resources you would need to use that feature as well, okay? So I'm going to save this layout, and I'm going to let's do something. Let's save it as unlinked, because now we've completed building the template, and we want to save it for future use with other sets of samples. So I'm going to call this our immunophenotyping tutorial template, and I'm going to save it as unlinked here. Okay, and so that's important. That's what's going to allow us to save it like an empty layout with no plots loaded. All right, and now when I close and reopen it from my recent docs here, you see that it's come up empty. So it's kind of similar to what we did at the beginning of the webinar, but now the layout is totally complete, and I can add in my files into the data list. Okay, and now we can load up the plots just by double-clicking on the file, the first file, and clicking OK. And now we're basically ready to go. So all of the files are here. Okay. So you can basically see all of them. And if we were to rerun the batch, everything would be all set. We don't have to set up the batch actions or anything. So there's no recreating the wheel there with FCS Express. Okay. So does anybody have any questions about that? Okay. So hopefully what you've seen today is that it's easy to load a layout with data files. Inserting plots and gates is very straightforward. There's a lot of drag and drop. And creating default and custom defined statistics is easy as well. Batch processing to programs outside of FCS Express can be done with a single click, and then you can easily reuse that layout as a template. So if there are any questions, I will hang out on the line for a little bit longer. Both Tanya and I are ready to answer any questions you have about anything we covered today. But if not, thanks so much for joining us, and we hope that we'll see you next time.